Hi, I'm Peter, and today I'll be teaching you about Gregor Mendel's work on inheritance. By the end of this lesson, you should understand 1. Mendel and his rules, 2. Mendel's experiments, and 3. How we can explain Mendel's results using modern biology. In 1865, Gregor Mendel published groundbreaking research about how genes are inherited. These rules are now named Mendelian genetics after him and lay the foundation for modern biology. Let's break it down. Mendel was a monk who lived in what is now known as the Czech Republic. After becoming interested in inheritance, which is how traits are passed down from one generation to the next, he carried out hundreds of breeding experiments using pea plants. He discovered that for every feature or trait that he investigated, a heritable unit or gene was passed from one generation to the next, which caused that particular feature or trait. Each of these heritable units or genes can have different forms, which we now call alleles. Each individual must have 2 alternative forms or alleles per trait. Gametes only contain 1 allele for each gene, and finally, while every individual has 2 alleles or versions for each gene or trait, one can be dominant over the other. That is to say that if the individual has 2 different versions of the same gene, only one may be observable as a trait. Using these ideas, Mendel was able to successfully predict the outcomes of his own crossbreeding plants, i.e. combining their gametes and observing the features or traits of the produced daughter plant. So let's take a look at these experiments. When observing pea plants, Mendel noticed that many features of them had 2 distinct forms. For example, plants were either very tall or very short. They either had purple flowers or white flowers. They either had green or yellow pods, and so on. What was striking to him was that there was no intermediate, that is to say that plants either had one form of the feature or another. For example, there were no medium-sized plants. So, he decided to breed a bunch of tall plants with a bunch of short plants to see what would happen. We can call the first generation P, for parents. He allowed this P generation to self-pollinate or asexually reproduce to form the F1 generation using their own gametes. He then crossbred different daughter cells from the F1 generation to produce the F2 generation. His results were very interesting. As you can see, all of the F1 generation plants were tall, but there was always a 3-to-1 ratio of tall to short plants in F2. And remember, this was done using hundreds of plants, so there wasn't always an exact 3-to-1 ratio. Mendel wasn't able to explain his results as he had no knowledge of genes, alleles, or chromosomes. However, we can now explain his results using our own understanding of these topics. So let's explain his results. We first need to understand some principles in order to explain them. Each feature, height in this case, is controlled by what we call a gene, which is found on a chromosome. Each body cell contains 2 copies of each chromosome, and therefore 2 copies of each gene. However, gametes, for example pollen or ovum, or sperm cells or egg cells in humans, only contain 1 copy of each chromosome. There are 2 versions of each gene. We call these alleles. For example, the gene for height has 2 alleles in pea plants, tall or short. One allele is dominant over the other allele, which is recessive. This means that if the individual contains 2 different alleles on each copy of a chromosome, only the dominant one will be shown in the appearance of the organism. However, when the individual contains 2 recessive alleles, the recessive alleles version of the gene will be shown in the appearance of the organism. That was quite wordy. To simplify things, we'll use the term genotype to describe the alleles for a gene which an individual has, and the phenotype to describe the form of the feature which is shown in the appearance of the organism. Usually we use a capital letter to show an allele is dominant and a lowercase letter to show it is recessive. Let's apply this to the height feature. Big T means tall allele and small t means short allele. Therefore, an individual's genotype is big T, it will be tall. If it is big T, or little t, big T, it will still be tall because the dominant tall allele will present itself, or be expressed, as we say, whereas the recessive allele will not be expressed. However, as you may have guessed, if an individual's genotype is little t, it will be short because there are no dominant alleles. Therefore, we say that the phenotype is short. Remember when I said that gametes only have one allele? That's very important when we look at crossbreeding. Half of an individual's gametes will contain one allele, and the other half will contain the other allele. For example, if their genotype is big T little t, half of their gametes will have the allele big T, and the other half will be small t. When sexual reproduction occurs, so when different individuals are crossbred, their daughter cells will contain one allele from each gene from each parent. We can represent this with a Punnett square. We can use a Punnett square to predict the likelihood of the daughter cell having different genotypes and therefore phenotypes. We put the male's gamete on the left and the female gamete on the top. Then we fill in each of the squares with the allele on the female gamete side and the male gamete side. Then we can count how many of each genotype there are. In this case, there is one big T, two big T little t, and one small t. Therefore, there will be a 3-to-1 ratio of big plants to small plants, as, if you can remember, big T, and also big T little t genotypes have the tall phenotype. However, little t has the dwarf or small plant, short plant phenotype. And this is the ratio of big plants to small plants that you will get and that Mendel observed if you crossbreed the parents an infinite amount of times. As this is random, there won't be an exact 3-to-1 ratio, as I mentioned before, but the 3-to-1 ratio predicts the likelihood of a plant being tall or short each time, in the same way that if you roll a dice, you have a 1/6 chance of rolling a 6 each time.